All the king's men. Schwann cells. Schwann cells, or neurolamocytes, originate from neural crest cells, going through several stages of development: boundary cap cells, Schwann cell precursors, immature Schwann cells, and mature Schwann cells. Schwann cell precursors are cells with processes that co-migrate with axons in the developing PNS and divide on their surfaces. Due to this mechanism, Schwann cell precursors are distributed throughout the body in parallel with the development of innervation. Schwann cell precursors closely interact with axons in the newly formed nerves, forming compact structures lacking basal laminae, blood vessels, and connective tissue. Moreover, contact with axons and molecular signals received from them are necessary for the survival of Schwann cell precursors. Influenced by several signals, the population of Schwann cell precursors gradually turns into immature Schwann cells. The main phenotypic differences between Schwann cell precursors and immature Schwann cells are as follows. The survival of immature Schwann cells in tissue culture does not depend on the presence of axons. Apoptosis is inhibited by autocrine mechanisms. In immature Schwann cells compared with Schwann cell precursors, the expression of S100 and glial fibrillary acidic protein is significantly increased. Interacting with axons, immature Schwann cells form basal laminae. Moreover, at this stage, the developing nerves already contain connective tissue and blood vessels. In the perinatal period, immature Schwann cells provide the so-called radial sorting. Schwann cells penetrate the nerve bundles and subdivide axons into groups surrounded by a family of 3 to 8 Schwann cells that form a common basal lamina. Then, Schwann cells' lamellipodia-like processes interact with large caliber axons, 1 micrometer or more, and segregate them to the periphery of the bundle. At this stage, immature Schwann cells divide, still within a common basal lamina, and daughter Schwann cells remain associated with axons and then separate them from the bundle to form their own basal laminae. As a result, the ratio between immature Schwann cells and axons of large diameter becomes 1:1. Only at this point can the Schwann cells, surrounded by their own basal laminae, turn on into promyelinating Schwann cells. They begin to myelinate axons and differentiate into myelinating Schwann cells, which form the myelin sheath around large axons. Following the separation of all the large caliber axons, immature Schwann cells that embrace the remaining small caliber axons differentiate into non-myelinating or remoc Schwann cells. The process of myelination proceeds as follows. The axon fits in a groove, invagination of the plasma membrane, along the surface of the Schwann cell. The folds above the axon close and a mesaxon, consisting of a double plasma membrane, is formed. Then mesaxon lengthens, forming wraps around the axon. The wraps, lined tightly 1:1, displace the cytoplasm of the Schwann cell and push the cytoplasm to the periphery and to the axon. Thus, the Schwann cell cytoplasm remains only in the form of narrow spaces around the formed myelin sheath, periaxonally, in the perinodal region and the so-called myelin clefts. Unlike the inner cytoplasmic compartment, which is uniformly spaced, the outer cytoplasmic compartment is interrupted by periodic appositions between the axonal membrane and the outer wrap of myelin sheath. These appositions delineate a network of anastomosing cytoplasmic channels termed Cajal bands. The nucleus of the Schwann cell is located in the region of the abaxonal cytoplasm. The myelin sheath is not continuous. It consists of segments sequentially located along the axon and formed by individual Schwann cells. They are separated by nodes of Ranvier, where action potentials are generated. The gaps between adjacent interstitial segments are filled with microvilli of the neighboring Schwann cells. Perinodal sites are adjacent to the nodes on both sides of them, in which the myelin lamellae end in the form of finger-like structures. Myelin clefts often develop following the substantial formation of the myelin sheath and arise as extensions from the inner or outer collars of cytoplasm by circumferential invasion into a compact myelin sheath, indicating an active process of decompaction of the myelin lamellae. As in the CNS, each myelin sheath, if unwrapped, is a structure containing compact myelin surrounded by a continuous cytoplasmic rim. PNS myelin, as in the CNS, is rich in cholesterol, glyco and sphingolipids. The galactolipids found in PNS myelin are substantially the same as those in the CNS. However, some lipids, such as sulfated glucuronyl paraglobusid and its derivatives, are specific for PNS myelin. Axons of small diameter, up to 1 micrometer, do not undergo radial sorting, but gradually fit into grooves along the surface of immature Schwann cells, which turn into non-myelinating Schwann cells, forming further myelin-free nerve fibers. The terminal or perisynaptic Schwann cells surround the terminal axon branches and synaptic boutons in the neuromuscular synapses and are part of some sensitive nerve endings such as Pacinian and Meissner's corpuscles. Perisynaptic Schwann cells are covered with a basal lamina that merges with that one of the muscle fiber and motor end plate. The formation of myelin sheaths in the nerve fibers of the PNS is the main function of myelinating Schwann cells. This process, as with the formation of myelin sheaths in the CNS, depends on the number of signals received from axons. At the same time, axons depend on trophic factors produced by Schwann cells. Other functions of Schwann cells: maintaining axon structure and vitality. Schwann cells mechanically support axons, provide trophic support, support ionic homeostasis, and prevent the ephaptic transmission of impulses between axons. The formation of nerve endings. Schwann cell precursors in large quantities are found in the area of nerve endings formation, creating a kind of matrix for axons growing here. This indicates a close interaction between neurons and glia and is important for the normal terminal branching of axons and their interaction with targets. The absence of Schwann cell precursors in special model experiments did not affect axon growth in the area of future nerve trunks. However, it led to abnormal sprouting in the area of nerve endings. The functioning and restoration of nerve endings. Terminal Schwann cells play an important role in the function of synapses, responding to nerve activity by increasing intracellular calcium. Terminal Schwann cells also play an important role in the restoration of neuromuscular endings, directing the growth of regenerating axons in the target region. The fundamental difference between mature Schwann cells and oligodendrocytes is their ability to turn into the repair Schwann cells after nerve trunk injury and to ensure axon regeneration. Saltatory conduction of nerve impulses, axon development and regulation of its diameter. These functions are similar to those of oligodendrocytes. Thank you for attention.